We talked about like chemiosmotic theory, where we saw that actually much of the ATP is produced by this theory, by chemiosmosis, in the electron transport system, where we said that as electrons are being transported from high energy levels to low energy levels, they lose energy, and that energy, you have the proton pumps within the inner membrane of the mitochondria. So there are those proton pumps that will pump these hydrogen ions from the matrix to the intermembrane space. So when you increase the concentration of hydrogen ions within this intermembrane space, they will tend to fall back, okay? But they can't cross this plasma membrane because it is hydrophobic, so it doesn't allow these polar molecules to cross. So as a result, they will need specific ion channels, okay, within the inner membrane where they will pass. And this ion channel, it is a potassium ion channel, but at the same time, it has a kinase activity, okay? So it is potassium ion ATP synthetase, whereby if it allows the inflow of hydrogen ions, since these hydrogen ions, there is a high electrochemical gradient between the intermembrane space and the inner mitochondrial the inner matrix, then that electrochemical gradient is going to be used to add an inorganic phosphate to ADP to produce ATP. And that was chemiosmosis, and we dissected it the other time. Okay? So now let's talk about the electron transport system. Those complex proteins that are found within the inner membrane and how they are able to do it. So within the inner membrane, let me use this pen. Within the inner membrane, we have there different protein complexes, okay, that are arranged in a series of decreasing energy. And they are these proteins that they have, obviously they have prosthetic groups within themselves, the non-protein components that get oxidized and reduced, okay? So when one is oxidized, then the other one is reduced, then the other one reduced, oxidized, okay? So they undergo oxidation reduction reactions as the electrons from Krebs cycle are flowing through these protein complexes, okay, at a series of decreasing energy, meaning that the electron that is flowing through these protein complexes, it will be flowing at decreasing energies, hence giving out energy. Now it is that energy that is given out by these protein complexes, okay, that will be used to pump these proton pumps to pump hydrogen ions, okay, from the matrix into the intermembrane space, hence increasing the hydrogen ions. And when you increase hydrogen ions, then chemiosmosis. So we have the very first complex 1, and this complex 1 is called NAD dehydrogenase, okay? The whole of the complex, okay, it's a complex protein. It is called NAD dehydrogenase or NADH dehydrogenase. And within this protein complex, you have there the prosthetic groups, the non-protein components. And in this case, okay, that's okay. And in this case, we have the flavin mononucleotide, okay? This is a prosthetic group okay, that is part of this protein complex. We are going to see the drawings. Okay, a moment, a moment. And then you have the iron sulfide, and the prosthetic group is iron. Now it is this iron, okay, that will be oxidized and reduced to channel the protein, sorry, the electron, at a series of decreasing energy. Then another complex protein within the plasma membrane is complex 2, and this one is made up of two. And the first one is the succinate coenzyme 2 reductase, okay, or you can call it coenzyme 2, coenzyme Q, or ubiquinol. Now this one is an enzyme. You remember in our Krebs cycle where we have a stage where the succinate is converted to fumarate. You remember that stage? Now this is the enzyme that catalyzes that reaction. Okay? Yes. So it catalyzes it by oxidizing the succinate, okay, to fumarate, and in the process it will take away the two hydrogens, and the two hydrogens will be accepted by the fat, okay, and they are channeled to cytochrome B, and the cytochrome B has these iron groups, okay, as their receptor molecules, then cytochrome BH, cytochrome BL, and finally the iron. Now all this, now it is still cytochrome B, whether BH, BL, it remains cytochrome B, okay, and then cytochrome C. We are going to see them, okay. Now these are the prosthetic groups within the protein complexes. Then with complex 3, this is cytochrome B1 complex, and then cytochrome C. They have the iron as their prosthetic groups, and they are these ions that get oxidized and reduced as electrons are traveled or are being shunned at decreasing energy levels. Then finally, complex 4, we have cytochrome oxidase. Now this is also an enzyme and the last electron acceptor as far as these protein complexes are concerned. And it has copper as its prosthetic group, or in the center, that will finally accept the electron before it is channeled to oxygen as the last electron acceptor. So this is basically what we are talking about. So that's basically what we are talking about. So we have this is the first complex, our first complex protein, the complex 1, okay, and then this is the second protein complex, okay, and then this is the third protein complex, and finally the cytochrome oxidase, the fourth protein complex, and the oxygen being an electron acceptor, the final electron acceptor. Actually, when we were studying respiration, I was disappointed with this oxygen. Let me first talk about that. I was disappointed with this oxygen. For me, all along I thought maybe the same way you heat charcoal, okay, to turn into carbon dioxide, the same way you can heat ethanol to produce carbon dioxide and water, is the same role of oxygen. But we studied respiration, and I was not seeing oxygen anywhere. But when I finally saw it here, I was like, really? Is this why oxygen is supposed to be here? But the answer is yeah. You must have a final electron acceptor that will turn the electrons and protons into a neutral molecule, which is water, so that you don't alter the pH and you don't oxidize or you don't reduce. You remove these electrons, okay, from these protein complexes to form a neutral molecule which can either be used by the body or you excrete, that is if you have excess water. Okay. And what is going to happen, you can see from this, these protein complexes, they are arranged in a series of decreasing energy. You can see. So as electrons are flowing through these protein complexes, they are losing energy. Okay? And it is that energy that will be used to pump the hydrogen ions. Okay? And I think we shall pump hydrogen ions at this, is it complex 2? And then complex 3. This complex 3. And then, yeah, at these two complexes. And I think also here at cytochrome oxidase. Now when you pump those hydrogen ions within the matrix, okay, within the matrix, no, within the intermembrane space, now there will be those hydrogen ions that will be used to produce energy by chemiosmosis. So let's see actually what happens. Let's see actually what happens. You remember the NAD? Okay, we produced NAD within the glycolysis. We produced NAD within the Krebs cycle. We produced NAD within the pyruvic acid activation stage. In all these stages, we are producing NAD. And we also produced FAD. Where is our FAD? FAD enters from here. Okay, actually from here, this complex too. Okay, this is our succinate dehydrogenase. That's complex 2, or you can say biquinone. So that's where it is going to enter from. Now, you produce FAD within the Krebs cycle. Now, all these ones, okay, all these complexes, they will be having high energy, and they are actually called the carrier molecules, okay, or carrier proteins. Actually, okay, carrier proteins, that they will carry your electrons together with protons from the cytoplasm, from the matrix into the electron transport system, where when they reach here, then your reduced NAD is going to be oxidized, okay, back into the oxidized form of NAD, okay, and the electrons, they will be taken up by the cytochrome B1, or by the cytochrome. And this cytochrome, it has iron as its prosthetic group. Okay, so when the iron receives the electron, now in stable form, it will be, the iron will exist as iron 3. Okay, iron 3. So when it receives the electron from NAD, like how you can see, it will be converted back to iron 2, as you can see. And then the proton is shunt, okay, from the matrix into the intermembrane space, this hydrogen ion. Okay, now this cytochrome B, obviously it will be unstable. Okay, it is unstable. That now the cytochrome C here, it will take up that electron. Okay, it will take up that electron and it becomes iron 2. Well, this is now the reduced form of cytochrome C, which is iron 2, as you can see. Now, as these electrons actually are being transported, now it has been fed on, it has been fed in at this stage, but right now, you see, it is at this stage. So there is a decrease in potential energy, okay, by that electron. So it is that energy, okay, that will be used to pump that proton from the matrix into the intermembrane space. And the same process happens, as you can see. Now this is iron 2. Now the cytochrome A with iron also, it will come and take up the electron, okay, from cytochrome C. And then the cytochrome A, it will be oxidized into, now this is the oxidized form, the reduced form, which is iron 2. Okay, and now the iron 2, which is in cytochrome A, it will channel the electrons. You can see in the whole of this complex. Finally, it will channel the electrons to the oxygen. And where is the oxygen? It is here. So the oxygen together with the hydrogen ions, okay, within the matrix, they will be taken up, they will combine to form water, which is a neutron molecule. So as you are doing all this, there will be energy that is lost. Okay? So now that energy that will be lost, it is the one that will be used to pump hydrogen ions. Now if you feed in NAD at this level, you pump hydrogen ions at this level, you feed in FAD at this level. Now this ubiquinone does not pump any hydrogen ions, so you pump hydrogen ions at this level. We are pumping hydrogen ions from the matrix, okay, from the matrix to the intermembrane space. Okay, you also pump hydrogen ions at this level. So as a result, with NAD, which is relatively at high energy levels, you pump 3 hydrogen ions. Okay, but with FAD, which is relatively at low energy levels, you pump just 2 hydrogen ions. So as a result, the net ATP that is formed by NAD, for each molecule of NAD, reduced NAD that is formed, it will produce 3 ATP molecules by chemiosmosis, and we explained chemiosmosis. Then for FAD, okay, for each FAD that is formed, okay, you form 2 ATP molecules. That is for each FAD that will be formed. So now, from that background, you can calculate how many ATP molecules will be formed per glucose level, per each glucose molecule. Should I give you time to calculate that? Should I? I think that's what we are going next. That's what we are going to talk about next. So description, we can go through that description. However, I've described almost everything. So in complex 1, which is called NAD reductase, the NAD donates the electrons to coenzyme flavin mononucleotide, which then passes the electrons to iron, okay, and the electrons lose some energy. The NAD is oxidized to this oxidized form, while flavin and iron are reduced to Fn and iron 2 respectively. Now, each electron is transferred with a proton. Now, as these electrons lose energy, okay, I talked about this, electrons from reduced iron are then passed to coenzyme Q. Okay, you saw the other complex too, that coenzyme Q. Then coenzyme Q is thus reduced while iron is oxidized, and then they go back to their natural form to be ready to accept another electron. So now, as this energy, it will be the one that will be used to pump the proton, so that in the end, you will fill the intermembrane space with the protons. And so what happens within complex 2, as you can see, in complex 2, that is succinyl or succinate dehydrogenase, electrons from FAD are passed to the iron and then to the coenzyme Q, which are then transferred to complex 3. FAD becomes oxidized to this oxidized form of FAD. So 4 protons are translocated across the inner mitochondrial membrane from the matrix to the intermembrane space. As you are doing that, then the proton gradient will later be used to generate ATP through oxidative phosphorylation by chemiosmosis. We saw it. During oxidation, FAD complex is bypassed because the complex 2 has only enough reducing potential to pass electrons to coenzyme Q. So all that then reduced coenzyme Q transfers electrons to complex 3, then passes through several cytochromes, those different cytochromes, and then iron proteins. During the process, iron is oxidized, is reduced from iron 3 to iron 2. The electrons lose additional energy and then passed on to cytochrome C. And the cytochrome C, this is passes, sorry for that, it passes electrons to complex 4, which is cytochrome oxidase, and finally passes the electrons to oxygen, which will accept the protons together with the electrons to form water, which is a neutral molecule. Complex 4 moves protons, producing a proton gradient, okay? And in the process, energy lost by the electrons in complex 1, complex 3, and complex 4 is used to pump protons into the intermembrane space, producing a proton gradient, which is used when protons flow down the concentration gradient through the channels in the stacked particles, okay? Then ATP synthase enzyme will be able to use the energy to generate ATP, and you produce enough energy. So I hope that one is okay. I hope it is explained, but in case we can repeat, it's okay. We can repeat, feel free. So the way that carried, you can see from NAD, but it is quick, eh? It's quick, eh? You can see. That's actually what happens. So now, so basically that is what respiration is about. That is a brief summary of respiration. So are we going to account from here? Yes, but before we account, I want you guys to try accounting for the ATP molecules, okay? And each one of you drops the number of ATP molecules that is produced. You'll be amazed that some of you have different answers, yet you are correct. So try to account and drop your answers. That ability to account, but this is a question. Account for I was almost saying the number. Account for the total number of ATP produced from one molecule of glucose. If the question is like that, the question wants you to describe the whole process of respiration, what we've been talking about. However, citing key steps in the formation of ATP, where NAD is going to be formed, or you can talk about the whole process, the entire process of respiration, but at the end you cite the key stages, okay? Where NAD is going to be formed, where ATP is going to be formed and by which process, where FAD is going to be formed, and then the NAD and the FAD will be transported to the Krebs side, to the electron transport system, where each will be broken and will be converted into 3 ATP molecules for the case of NAD, and for the case of FAD, each will be converted to 2, and you have your ATP. Do you have the answers in the chat box for the ATP molecules produced? Let me check. So I was amazed with your answers, and the fact that you guys could give some answers, then it shows that you can describe, okay? You can calculate the entire ATP molecules that are formed, okay? Which is good. Now let's see. The ATP that can be formed, it ranges. If you don't form 36, you will form 38. So if you have all these answers, you are correct. And I'm going to show you how it is going to be 36 and how it is going to be 38. Now, It's okay, let it stay there. Now, remember in glycolysis, we invested two ATP molecules. Okay? But when it's split into two, each the dihydroxyacetone phosphate and glyceraldehyde, phosphoglyceraldehyde 3 or glyceraldehyde 3 phosphate, each of the two was able to produce two ATP molecules. Okay? So 2 plus 2, that's how many? That's 4. Are we together? Yes. So those are the 4 ATP molecules. But remember, you have invested how many? 2. Now in pyruvic acid, we produced NAD. Okay? We produced NAD. And each molecule of NAD is able to produce how many? 3 ATP molecules. ATP molecules. Now, however, in this stage, okay, there are two ways. So there are two things. You can either produce NAD or FAD. Now, if you form NAD, that's when you produce 38 ATP molecules. At this stage, if the two electrons together with the protons are taken up by the FAD, then you produce 36 ATP molecules. But if the two ATP molecules, so if the two electrons, oh, another loss of connection? Oh my god. Oh, somebody has joined. Let me check. We were saying, sorry for that, guys. So we're saying with each glucose in glycolysis you invest 2, okay, but you produce 4 in glycolysis, the 4 ATP molecules, okay, but the net, obviously we shall get 2 minus the 4, and the net remains the 2 ATP molecules. Now in this the pyruvic acid activation stage, this process of converting pyruvic acid to acetyl-CoA, there is NAD that is produced. Now you can either produce NAD, okay, if the electrons are taken up by NAD, or the electrons can be taken up by the FAD. Okay? Now if the electrons are taken up by the NAD, that's when you produce the 38 ATP molecules. But if the electrons are taken up by the FAD, then that's when you produce 36 ATP molecules. Now, you form one NAD, but remember, from here, there are 2 cycles running. Okay? There are 2 cycles running because each glucose molecule is going to split, okay, to give dihydroxyacetone phosphate and glycerate 3 or glyceraldehyde 3 phosphate, not glycerate, okay, glyceraldehyde 3 phosphate. So there are 2 molecules running. So if we produce one NAD here or one FAD, then the one FAD will be times 2. Okay? Actually, this NAD is for glycolysis. Okay? Sorry for that. This is for glycolysis. I didn't. So we produce one NAD, but since there are 2 molecules running, then we shall multiply by 2. Okay? Here the same thing. We shall produce one NAD, the electrons in this stage. Okay? That stage of converting pyruvic acid into acetyl-CoA. It can be taken up by NAD or by FAD. So if they are taken up by NAD, then it will be times 2. Okay? And each NAD will generate a maximum of 3 ATP molecules. You remember that? Yes. So 3 times 2, that's a total of 6 electrons. So the 6 ATP molecules. Then within Krebs cycle, we shall produce 3 reduced NAD, but there are 2 cycles running. You remember that? Yes, that's pretty good. There are 2 cycles running. So it will be 3 times 2, which is 6. But each NAD can produce a maximum of how many? 3 ATP molecules. So that will be a total of 18 ATP molecules. Then with this, we shall form one FAD within the Krebs cycle. Then there are 2 molecules running, so that is 2 times the 1 FAD, that's 4, and each FAD is able to produce how many? 2 ATP molecules. So the total is going to be 4 for each FAD. Okay, but now there is one ATP molecule that is formed within the Krebs cycle, and the ATP molecule is formed by substrate level phosphorylation, but there are 2 molecules running, so that is the 2. Okay, so when we add, this is 4 plus the 4, that's 8, plus the 6, that's 14, plus 16, then plus 16 plus that is 34, plus 4, that is 38 ATP molecules. But you have used the 2, so if they want the net, then you subtract the net. Okay, so minus this net, then we shall produce a net of 36 ATP molecules. You use 2, you produce 36. So that's the principle of investing. It is even working here. Okay? You invest 2, you produce 36. But some of you are thinking that you don't even want to invest. So if you can't invest, then you can't produce anything. Okay? So basically that's how we can account for the ATP molecules that are produced within this. Any question for me? Maybe let me check the chat box for any queries of oxygen. Now what happens if oxygen is absent? What happens if you cannot supply enough oxygen, okay, to oxidize the food? Then your glucose will undergo what we call fermentation. And which type of fermentation? We have 2. We have alcoholic and then the lactic acid fermentation. So now it will depend on the type of organism. If it's you and other mammals, then you undergo, and most animals in fact, then you undergo lactic acid fermentation. Okay, lactic acid fermentation, where you produce lactic acid. But if it is the plants and then the, I think, fungi, then these fungi do undergo what we call alcoholic fermentation, where they will convert the pyruvic acid that is formed. It is the same glycolysis, but the pyruvate that is formed, it will be converted to it will be converted to alcohol. Okay, and actually people, the smart guys have utilized this. That's why if you have ever been to a brewing industry, or if you can brew locally, I have grandma who can do that. Whenever she wants to make some ethanol, she covers everything, like serious covering. Now why does she cover? To cut off oxygen. And then she adds yeast, so that the yeast undergoes what we call alcoholic fermentation. It undergoes normal glycolysis, then the pyruvic acid is converted to ethanol. and then she extracts, she filters, she separates ethanol from the other tonto by distillation. So what happens in absence of oxygen? It is simple logic. Fermentation. And fermentation is also a metabolic pathway that regenerates the oxidized form of NAD. Okay? By oxidizing the stockpiles of reduced NAD. Remember when you are sprinting, eh? Like, should I say Cheptegei? Or should I say any sprinter? The other one is a marathon. Even those people, those guys of marathon. You reach a level where the energy needed, where you need energy, too much of it, than the oxygen that you can supply to the respiring tissues. The rate at which you can supply the oxygen is way, way below than the energy that is needed. So the muscles resort to anaerobic respiration, whereby we have seen that they will undergo just glycolysis and use the two ATP molecules that are formed, just two. Okay? You invest two, you produce four, so you have a net of two ATP molecules. Just two. However, these two, they are robust, they are immediate. Okay? They are immediate, so the muscles, they will get energy as soon as they want it. But yes, it's good you are producing energy immediately, but however, you see, you are forming reduced NAD. Okay? And then the reduced NAD is going to accumulate within the muscles. Okay? And if they accumulate, then will the process of respiration run? I don't know. And if the process doesn't run, then what is going to happen next? Will you carry out respiration? I don't know. Okay? You won't carry out the respiration. So as a result, there is a need to regenerate back the oxidized form of NAD so that the process continues. And how do you regenerate it back? By fermentation. Whereby the NAD, okay, is going to be used to reduce the pyruvic acid, okay, into the lactic acid, okay, and the NAD itself is oxidized back into the oxidized form of NAD so that it is made available for the process to continue. Okay? Yes, so the electrons from NAD, from reduced NAD, are transferred to pyruvic acid, okay, or a molecule derived from pyruvate instead of electron to be shunt in the electron transport system. Depending, if it is alcoholic fermentation, then we shall see. If it is lactic acid fermentation, then the electrons are donated directly to NAD to form the lactic acid. And so you'll be able to regenerate back the oxidized form of NAD and you maintain the cycle. So basically this is what we are talking about. So maybe let me use this. So you have your glucose and you will oxidize the glucose, that is glycolysis, to form pyruvic acid. Now the pyruvic acid, if oxygen is there, then it will undergo this pyruvic acid activation stage and then citric acid cycle to the electron transport system where energy is going to be produced. and the final electron acceptor is going to be the oxygen, where it will be converted to water. But now, if oxygen is not enough, if oxygen is not sufficient, then you cannot undergo this. You cannot undergo this process. Okay? Yes, so what will you do? You will rechannel, you undergo fermentation, where the pyruvate, yes, there will be a lot of energy that is not, that won't be, this is still a high energy containing compound. It still have more hydrogens, but you have nothing to do, there is no oxygen. Okay, so you convert it back to lactic acid or ethanol. But the good thing is the lactic acid, if you're done with your exercise, you pay that oxygen debt, and the lactic acid can be converted back to pyruvic acid and it proceeds. Okay, and you respire and you produce the energy. But this fermentation is an immediate intervention, okay, to produce that robust energy that will be needed in respiration. And so there are two types. Number one, we have alcoholic fermentation. Here we form alcohol. And number two, we have the lactic acid fermentation. And what's up with alcoholic fermentation? The pyruvic acid that is formed is going to undergo what we call decarboxylation. It is an acid, okay? So it will be decarboxylated, okay, to form a two-carbon compound, acetaldehyde. Okay, this is acetaldehyde, CH3, and then CH. Then you have this, and then the H. So that is your acetaldehyde. So you decarboxylate the pyruvic acid. You remove the carbon dioxide. So this is your carboxylic, your pyruvic acid. This one, you take away this. Okay, and if you take away this, then you remain with acetaldehyde. Now, from chemistry, remember, when you have this and you want, now this is organic chemistry. When you have this and you want to convert it to ethanol, what do? It is CH3. You use a reducing agent. And in chemistry, what do we use? Don't we use lithium, aluminium hydride in presence of dry ether? This is chemistry. If you are not a chemistry student, sorry for this. So we don't we use lithium, aluminium hydride, and we convert it to this? Yes, that's brilliant. So also here, instead of using lithium, aluminium hydride, we don't have it. That is now for the laboratory setup. Here we shall use our NAD, our reduced NAD. Okay? We shall use our reduced NAD to reduce, okay, to reduce our ethanol, okay, to ethanol. And then the NAD itself, it will be oxidized to the oxidized form of NAD. Okay? And it will be ready to accept more electrons and more protons. And the cycle continues. Oh, sorry for that. And the cycle continues. And you can see that. So acetaldehyde is then reduced to the ethanol, and that one, NAD, enables the continuation of glycolysis. And alcoholic fermentation occurs in some bacteria, yeast, and plants. They produce ethanol. So if you like ethanol, if you like H functional group, this is what you use. Okay? And the ethanol is not useful in your body. It is not useful in your body. Once you take ethanol, it will undergo alcoholic degradation, where it will be degraded, okay, and removed out of your body because it doesn't have any nutritious value. Okay? But in the process, you produce a lot of NAD, reduced NAD. Okay? In the process of degrading the NAD, the alcohol, you produce a lot of reduced NAD in the liver. And now you see this, you want to respire, but NAD is in this format. Okay? But yet you want this oxidized form of NAD. So you have nothing to do. So as a result, respiration won't occur, and so you won't have the energy, and that's the feeling you will feel after taking your OH functional group. In the morning, you feel like you have hangover. Hangover. What is that hangover? It's because the body does not have energy. And if the body doesn't have energy, then you feel weak. The little energy that is available, it is reserved for the vital organs. The heart cannot stop pumping. The brain must have that constant supply of energy. The chest, okay, it keeps on moving up and down. Respiration, inspiration should keep taking place. So you reserve the little energy for the vital organs. And as a result, the muscles feel they lack energy, and you feel that general body weakness. Oh, why body weakness? And what is wrong? So you feel weak because the body does not have energy, and that's the hangover. Why? Because all the NAD is in this reduced form. It has been used to oxidize the ethanol. Okay? Now, how about with lactic acid fermentation? Now this one, there is no decarboxylation. Okay? There is no decarboxylation. And in fact, with anaerobic respiration, no carbon dioxide is produced in mammals, because the pyruvic acid is directly reduced by the NADH to form the lactic acid. Okay? And there is no carbon dioxide is produced. However, since this is an acid, lactic acid, you remember it in chemistry. It is, if we try to sketch how it was seen here, what? So this is H, you have the OH here, and then the carboxyl group. So this is the lactic acid. Okay? So this is your lactic acid. Now, because it's an acid, because it has a carboxyl group, it can lose the hydrogen ions, which will alter the pH. So that's why as soon as you finish, then you undergo the oxygen depth to break down the lactic acid. Okay? But it can also be converted back to pyruvic acid. It's a matter of reducing this. Reducing, you oxidize. I think you oxidize this. Okay? By taking away this hydrogen by the NAD. Okay? And you have your pyruvic acid. Your pyruvic acid. And then it proceeds to the electron transport system. So it is that simple. However, if you leave it to accumulate, then it will alter the pH in the skeletal muscles. And if you alter the pH, and then you know what will happen. Okay? You disrupt, you entangle, you disable the physiology in cells. So it is carried out by certain fungi and bacteria, and during the formation of yogurt and cheese. It also occurs during oxygen scarcity in human skeletal muscles, during sprinting. They will send bolts. You can, oh, it is not you, it is someone. But you can also compare the sprinters. Are you aware that somebody who can sprint cannot run a marathon? And are you aware that someone who can run a marathon cannot sprint? Now you can find out the different adaptations of these two. Okay? Why Eliud Kipchoge can run 40 kilometers, but you cannot run from goal to goal. he cannot participate in such. And this guy, the Bolt, Usain Bolt, I think you know him. If you are a good sportsman, you know Usain Bolt. He holds the record. Imagine running from goal to goal, running 100 meters in is it 9 or 8? I think it was 9.56 seconds. Was it 9 or 8? You find out. 100 meters in 9.56 seconds. I think it's 5.4. Meaning, in every second, the guy was running 10 meters. Can you imagine? They're meant to be like 1, 2. The guy has run 10 meters. 1, 2, 3. The guy has run 30 something meters. So a sprinter, the two are adapted differently. Okay? And it can be some good question for you to find out. So try to do some reading. Okay? And find out. So the lactic acid is gradually carried away by blood to the liver, where it is converted back to pyruvate. Yes, what I've been talking about. By the liver cells. Okay? And then they are respired. If ATP is abundant, then pyruvic and lactate can be used as a substrate in synthesis of glucose. If you have enough ATP, if you convert back your pyruvic acid, then you can use these ones to form the glucose back in the liver cells. But if the ATP is not enough, if you still want the ATP in your body, then you can still break down the lactic acid into ATP, okay? And you produce your normal ATP. And you flourish. I hope that is okay. So this is basically what we are talking about. So this is your glucose, and the glucose is converted to pyruvic acid. Okay? This is glycolysis, and you have your pyruvic acid. Then the pyruvic acid will be converted to lactic acid. Okay? Now, this is normal chemistry. If you know organic chemistry, this is normal chemistry. Where this aldehyde group, this one, this carbonyl group, it will be reduced to an alcohol. If you know your chemistry, then you are good. And what does? It is the NAD that does that. Okay? And you have your lactic acid. However, when you're done with the exercise, then you convert back to pyruvic acid, and then you proceed the Krebs cycle and then electron transport system, and you have your energy. But if you don't have enough, if the energy is enough, if energy is more than enough, then you convert back the pyruvic acid to glucose. Okay? Yes, and you store the glucose. So it is as simple as that. So let me. Okay, let's talk about the economic importance of anaerobic respiration. Like I said, it is these normal things. Okay? The breweries, the brewing industries, they have used this anaerobic respiration to make money. I hope these guys have made a lot of money. A lot of money. Okay? And the money they are making, it is from this anaerobic respiration, where they're able to convert sugars into ethanol, and then they use ethanol to make wine. For example, you make wine from grapes, some good wine. And grapes, that is purely maltose, okay, and glucose. So some good wine, but it's all ethanol, nothing different, just the flavor changes. Beer, it is ethanol, nothing, and the flavor, but it's ethanol. And then spirits, the spirit of Uganda. Do you know it, guys? the spirit of Uganda. Do you like it? Yes. Bakeries. These guys that do bakery, they have also used this to make the good luxuries, luxurious breads, cakes, and so on. So we ferment starch using yeast. Okay. For example, you buy a piece of bread, when you buy bread and you find there are some patches in between. Those patches are due to sodium carbon. I think it is the carbon dioxide, and the carbon dioxide is produced from that bicarbonate that is added. But alternatively, if you don't want to use that, you can also add yeast. Okay, and then the yeast undergoes fermentation. Since it is alcoholic fermentation, you produce carbon dioxide. Then the carbon dioxide that is produced, it's the one that raises the bread. Okay, so those are the patches that you see. Okay, then manufacture of milk, such as the sour milk. I don't know how that one tastes, but automatically it is sour. So, because of this word, now me I hate sour things, so I can't take it. Yogurt. I hope you guys here take yogurt. And cheese. I hope you guys here take cheese. And all these ones, they are products of anaerobic respiration. And finally, food industries manufacture organic acids such as citric acids, oxalic acids, and butyric acids. Now these are derivatives of lactic acid, I think, that is produced. So you can use that and you can use the lactic acid that is produced to form these acids, and so on and so on and so on. So I don't